During this chapter, we are going to go over the basic structure of the brain, and this is part 1 of the 2-part lecture about the brain. So just a general outline, as we go through this chapter, the first thing we're going to do is look at the cranial meninges and talk about the cerebrospinal fluid, followed by the structure of the brain itself. So we're going to talk about the different structures and the different parts of the brain, followed by how they are grouped. And then we're going to look at the different centers in the brain. So brain being part of the central nervous system, we have sensory areas, we have association areas, as well as motor areas in the brain. So in the last part of the chapter, which will be included in the part 2 section, we will talk about the different centers in the brain. So to start the brain off, what you're looking at here is a sagittal section medial view of the brain. So cutting right along the sagittal suture, and you're looking at it from the medial side. So anytime you want to refer to the front of the brain, so the side of the brain that's oriented towards the frontal bone, that end is referred to as the rostral end. The back side of the brain, the one that is located towards the occipital bone, is referred to as the caudal end. So you have the rostral end and the caudal end. Now, looking at the major sections of the brain, the largest part of the brain is the cerebrum. So this whole thing, the noodly appearance right here, the noodles, that is the cerebrum. It's the largest section of the brain. Now, the brain stem is right here. So this thing right here, that is the brain stem. And it's divided into 3 parts, which we will be looking at in just a little bit. The third section of the brain is the cerebellum, this thing right here, and followed by the diencephalon. So the diencephalon is the central region of the brain. So this guy right here is the diencephalon. So the biggest section of the brain are the noodles, which is the cerebrum. Then you have the cerebellum, the brain stem, and the diencephalon, which is the central and the deepest part of the brain. Now, the front of the brain towards the frontal lobe is called the rostral end. The back towards the occipital bone is called the caudal end. Now, in the cerebrum, we have a whole bunch of bumps and indents, and bumps and indents. So that is what the folds and the indents are, the ones that give the cerebrum its characteristic noodly appearance. Now, anytime that you have a fold or a bump, it is referred to as a gyrus, G-Y-R-U-S, singular. Several of them together, the plural form is gyri. So gyrus or gyri are folds or bumps in the tissue. The indents, or the grooves, are called sulcus or sulci. So we have gyri and sulci. Now the biggest line that you see right here in the cerebrum is this. And that right there is called the longitudinal fissure. So this is the superior view of the cerebrum. The longitudinal fissure divides the cerebrum into the right hemisphere and the left hemisphere. So this is a deep cut. So it's not a sulcus, it's a fissure because it goes through and through. Now we have a lot of gyri, a lot of sulci all along the surface of the cerebrum. There's one that clearly demarcates the front of the brain to the back of the brain. And this sulcus right here goes like this and like that. That is referred to as the central sulcus. So you have the longitudinal fissure that divides the brain into the left and the right hemispheres. The central sulcus divides the brain into frontal lobes and the parietal lobes. So this is the right frontal lobe, this is the left frontal lobe, and then you have the 2 parietal lobes. Now you have another sulcus right here that divides the cerebrum into parietal lobes and occipital lobes. So think about the lobes as they are positioned behind the bones of the skull. So frontal lobes are behind the frontal bones. Parietal lobes are behind the parietal bones, and occipital lobes are behind the occipital bones. So you know now what rostral is, what caudal is, what gyri are, what sulci are. You know the longitudinal fissure, the central sulcus. Now you have another sulcus which is viewable from the lateral view, and it is called the lateral sulcus. It's right over here. And the lateral sulcus divides the cerebrum into the temporal lobe. So the temporal lobe is present right behind the temporal bones. We've already talked about this a little bit before. The location and the positioning of the gray matter versus the white matter. Now, in the brain, unlike the spinal cord, the gray matter is towards the periphery, or it's more superficial. The gray matter is gray because it has synapses, it has unmyelinated axons, it has somas or cytons of nerve cells, as well as dendrites. These regions are very rich in synapses, and they appear gray visually because of the lack of myelin, which is a lipid-based substance. Now, in the brain, the gray area is superficial, and it is referred to as the cerebral cortex. Now, the thing is, the observations are that the more cerebral cortex, or the more gray matter you have, the higher IQ you seem to have. So Albert Einstein had quite a bit of gray matter in his brain. Now, the white matter is located deeper within the brain, so not superficial. However, scattered in that white matter, we have certain regions of gray matter. So you have certain gray areas deep inside the brain, and they're referred to as the nuclei of the brain. Do not confuse this with the nucleus of the cell. The nuclei of the brain are deep regions within the cerebrum that are gray matter, and they're surrounded on all sides by white matter. Now, the white matter is white visually because it contains myelin, and that comes from the myelin sheath that covers the axons of the nerve cells, or the neurons. The next aspect that we're going to look at are the cranial meninges. Meninges, or meninx in singular, are the layers that form around the brain and the spinal cord, and they're for protective reasons. So we're going to look, in the spinal cord chapter, we looked at the three meninges from the most superficial to the deepest. So we talked about how the dura mater is the most superficial meninx, followed by the arachnoid matter, followed by the pia mater. So this is going to continue when you get into the cranium as well. So the spinal meninges and the cranial meninges are very, very similar with some minor differences. The outermost meninx of the cranium is still called the dura mater. It is thick, it is papery, and it is protective. Now, in the cranium, we do not have the epidural space that you see around the spinal cord. So the absence of the epidural space is difference number one. Now, this dura mater in the brain is composed of two layers. The outer one is called the periosteal layer. If you remember, the periosteum was the sheet of connective tissue that surrounded the bone. So the periosteal layer is basically the layer of dura that runs right along the periosteal layer of the skull bone. The inner layer is called the meningeal layer, and this is the one that continues with the meningeal layer, the dura mater of the spinal cord, with the epidural space not being present. So let's take a look at, the next meninx. The next meninx is the arachnoid mater. It is the same. It continues along the spinal meninx, which is the arachnoid mater as well. It does have the subarachnoid space, which is continuous with that of the spinal cord, and it also contains cerebrospinal fluid. The difference here is the presence of the arachnoid villi. The arachnoid villi are little finger-like projections that jut out into the subarachnoid space, and they allow for the wastes and toxins and gases to be able to be absorbed from the circulating cerebrospinal fluid. So this image is nice because it explains all of the different layers pretty good. This is the skin of the skull with its adipose tissue. This is the bone right here. Now you see in the green layer you have two sublayers. So the one oriented towards the bone is the periosteal layer, and the inner one is the meningeal layer. Now the red layer is the arachnoid matter, and you see that these finger-like projections right here are the arachnoid villi, and they help to absorb stuff out of the cerebrospinal fluid. And this very thin red layer over here is the pia mater. Now typically, because of the tight junctions in the endothelium, which line our blood vessels, we have a pretty good blood-brain barrier. However, you can have pathogens that can defy that blood-brain barrier and cause the inflammation of the cranial meninges, as well as the spinal meninges, and that is referred to as meningitis. Now, meningitis, or the inflammation of the meninges, can be because of a lot of different reasons. It can be bacterial infections, it could be viral infections, or rarely it could be helminthic infections as well. And it's a fast progressing disease. So bacterial or viral meningitis typically progresses very fast. So a person could be completely asymptomatic but develop a high fever, stiffness of the muscles, headache that could lead to a coma. And the only way to kind of diagnose it, aside from symptomatic diagnosis, a complete diagnosis requires looking at the pathology of the cerebrospinal fluid. And the way to extract cerebrospinal fluid is via a lumbar tap. A lumbar tap is when a needle is inserted into the subarachnoid space, and this is usually done in the lumbar region, and the needle is inserted right between the intervertebral disc all the way down to the subarachnoid space. A little bit of CSF is aspirated out and sent to the lab for testing. Now, adults are usually less susceptible to meningitis, but children and young adults, however, are at risk for meningitis. And there is a vaccination for children and adults. It's called Menactra, and it has shown pretty good results in preventing bacterial and viral meningitis. The next thing we're going to look at is the blood supply to the brain. Now, the brain, being just 2% of the body weight, takes about 20% of the oxygen because of its functions. It also requires a lot of glucose. And we have arteries and veins that carry this 20% of oxygen and glucose to the brain. Now, 4 minutes, an average of 4 minutes without oxygen to the brain or glucose to the brain can cause irreversible brain damage. Now, if you've been sitting for a long time, or if you've been in one position for a long time and you stand up or change positions quickly, you tend to feel what we refer to as lightheadedness or dizziness. And that is because the brain can sense that lack of blood flow, and it can cause disorientation. A very important part to note here is the blood-brain barrier. The blood-brain barrier is basically the characteristic of a few things. One, the astrocytes, which are the glial cells of the CNS, participate in blood-brain barrier. The second one is the tight junctions in the endothelial cells. So endothelium is the epithelium that is present inside of our blood vessels, if you recall from our tissues chapter. Now, these endothelial cells have tight junctions that don't allow much to pass in between. So that is the blood-brain barrier. However, the blood-brain barrier isn't completely impermeable. Gases, substances that are usually lipid-soluble, can make their way into the brain. For example, alcohol, nicotine, which is from cigarettes, THC from marijuana, oxygen, carbon dioxide, absolutely necessary for survival, as well as certain over-the-counter medications. One example of that is Benadryl. Now, we've talked about the cerebrospinal fluid, and you know that the cerebrospinal fluid is present between the subarachnoid space, and that is the case around the brain superficially. But we have hollow spaces inside, deep inside of the brain, that flow cerebrospinal fluid through it as well. And these hollow spaces inside the brain are called ventricles, or brain ventricles. So a brain ventricle is nothing but a hollow space inside the brain with cerebrospinal fluid in it. So I'm going to explain all of these ventricles from this image right here. So let's take a look at the ventricles. Now these two ventricles right here, let me change the pen color. Okay, 1 and 2. Now these are horseshoe shaped, so they're U-shaped ventricles present in either hemisphere. So you have one in the left hemisphere, one in the right hemisphere. And those are referred to as the lateral ventricles. So you have two lateral ventricles, one in the left and one in the right hemisphere. Now on the inferior side of this horseshoe right here, you have a connection. And that connection is called the interventricular foramen. Inter for between, ventricles for the ventricles. So interventricular foramen is a junction between the lateral ventricles, which are referred to as ventricle 1 and ventricle 2. And this is the third ventricle right here. So the interventricular foramen is connecting the lateral ventricles to the third ventricle. Now, there is a fourth ventricle as well. Let's take a look at where the fourth ventricle is. It is right over here. So you have lateral ventricles, you have the third ventricle, and you have the fourth ventricle. Now you have a long tube that connects the third ventricle to the fourth ventricle. So it goes right here. And that is called the cerebral aqueduct. So the cerebral aqueduct is connecting the third ventricle to the fourth ventricle. Now, the fourth ventricle goes back down into the central canal of the spinal cord. And there are two offshoots to it. Those two offshoots are one on the medial side and one on the lateral side. So the one on the medial side is called the median aperture. The one on the lateral side is called the lateral aperture. So let's recap the ventricles of the brain. So we have 4 ventricles. The first 2 are referred to as lateral ventricles, and they are U-shaped and present one in each cerebral hemisphere. The lateral ventricles are connected to the third ventricle by the interventricular foramen. The third ventricle is connected to the fourth ventricle by the cerebral aqueduct. The fourth ventricle travels downwards into the spinal cord and turns into that central canal that is present right in the center of the spinal cord. The 2 offshoots to this are the median and the lateral apertures. Now, all the ventricles contain something called choroid plexuses. Now, choroid plexuses are rich in ependymal cells. If you remember from our nervous tissue chapter, ependymal cells are glial cells that make cerebrospinal fluid. So these plexuses are choroid plexuses, and they are rich in ependymal cells to create cerebrospinal fluid, as well as blood capillaries. So what we're going to do next is take a look at, this is a frontal section, so you can see the 2 lateral ventricles, 1 and 2. You have the third ventricle, whoops, this is the third ventricle, this is the fourth ventricle, and this is the central canal right over here. You can also see the median and the lateral apertures. Now, cerebrospinal fluid is a derivative of blood. It is a colorless liquid which is sterile in normal conditions, unless there is an infection of the cerebrospinal fluid causing meningitis. We make about half a liter, or 500 milliliters, of cerebrospinal fluid a day. And cerebrospinal fluid, this system is kind of connected with the venous sinuses. The venous system, the circulatory system, has the arterial system and the venous system. So the venous system is what is going to carry carbon dioxide back to the heart. So reabsorption of cerebrospinal fluid happens via the venous sinuses that we're going to look at. So we create 500 milliliters of cerebrospinal fluid a day, and we have to also absorb about that much every day. So unable to reabsorb or drain cerebrospinal fluid is a condition called hydrocephaly. So we have to get rid of enough and make enough. So if there's a lack of balance between the two, there's a problem. So put a star mark next to this image. This is an important image, especially the flow chart. Not the image in general, because the image is a little bit overwhelming, but the flow chart. So here's the flow chart that shows you the direction of flow of the cerebrospinal fluid. So you have blood, the heart is pumping the blood, the lungs are getting the oxygen in, the oxygen is diffusing into our bloodstream, and the heart is pumping through the arterial system into the fourth ventricle. Whoops. So it goes into the fourth ventricle, it goes into the third ventricle, goes into the lateral ventricle. You know that all of these ventricles, so through the arterial system, it goes to the choroid plexuses, which are present in these ventricles. So from the choroid plexus, this blood, in conjunction with the ependymal cells, is being converted into cerebrospinal fluid. So all of the ventricles are capable of converting blood into cerebrospinal fluid. So the lateral ventricle is doing it, the third ventricles and the fourth ventricles are all doing it simultaneously. Now whatever is produced in the lateral ventricles goes through the intraventricular foramen into the third ventricle. Whatever gets produced in the third ventricle goes through the cerebral aqueduct to the fourth ventricle. And from the fourth ventricle, it can either, 1, flow down into the central canal, 2, it goes through the lateral and median apertures and into the subarachnoid space. Now, if you remember, the subarachnoid space has arachnoid villi. So the arachnoid villi are reabsorbing the cerebrospinal fluid, and via the venous system, taking it back to the heart and then eventually to the lungs. So let's go over this one more time. So when you inhale, oxygen is coming into your lungs, oxygen is getting diffused from the lungs into the bloodstream. So from the bloodstream, the heart is pumping this oxygenated blood to the ventricles, to all the ventricles, lateral, third, and fourth. So from the ventricles, the ependymal cells are removing the red blood cells and all of that stuff and converting the blood into the cerebrospinal fluid. So whatever cerebrospinal fluid, it flows through that closed circuit. So from the lateral ventricle to the third ventricle via the intraventricular foramen, from the third ventricle to the fourth ventricle via the cerebral aqueduct, and then from the lateral and the median apertures to the subarachnoid space. From the subarachnoid space, it can get reabsorbed via the the arachnoid villi. Now this is deoxygenated stuff at this point, and it is taken back via the venous system back to the heart and to the lungs. Now cerebrospinal fluid is extremely important. 1, it is a shock absorber, so it cushions the brain from hitting the inside of the skull, so it prevents impact. Now the density of the brain and the density of the cerebrospinal fluid are the same. So the brain, if you take a beaker of cerebrospinal fluid and put a brain in it, the brain is neither going to float to the top nor is it going to sink to the bottom, and that is because they are similar in density. Now, what is holding the brain in place inside the cranium is specialized fibroblasts. If you think of fibroblasts, fibrous connective tissue, we're talking collagen fibers. So the arachnoid matter is rich in these special fibroblasts that are kind of holding or suspending the brain in its proper place. Now the brain cannot afford to put its weight down on the floor of the cranium either, because it would kill the tissue that is on that floor of the cranium. Now, cerebrospinal fluid also plays a role in homeostasis, and it regulates the pH. So anytime you have low pH of cerebrospinal fluid, so low pH means more acidic, and that more acidity is due to the high concentration of hydrogen ions. Now, that means that your brain needs oxygen, or is high in carbon dioxide. So anytime the pH of the cerebrospinal fluid goes down, ventilation or breath rate increases. Now, anytime you have too much oxygen in your blood, and that causes the hydroxide ions to increase and the low hydrogen ion concentration, and that causes the pH to go up. And whenever the pH goes up, that is against homeostasis as well, so it decreases or slows down breath rate. So now we're going to look at the structure of the brain stem. The brain stem is divided into three regions. The inferiormost is the medulla oblongata, the middle one is the pons, and the most superior part of the brain stem is the midbrain. And we have a network of gray matter called the reticular formation that kind of connects these regions with the rest of the brain. So let's take a look right here. This red arrow is pointing to the medulla oblongata. Medulla oblongata is continuous or rises from the spinal cord, and this is present right at that foramen magnum. So everybody remembers the foramen magnum. So that is where the foramen magnum is located. So right at that junction is where the medulla oblongata is located. Towards its anterior side, this is the anterior side, it has pyramid-shaped structures that contain corticospinal tracts. Now, corticospinal tract, if you look at the name, it's going from the cortex of the brain down towards the spinal cord. So corticospinal tracts are descending tracts. So the pyramid-like structures on the medulla oblongata are home to descending or corticospinal tracts, which are nothing but motor tracts. Now let's look at the sensory functions and the motor functions of the medulla oblongata. One, it contains ascending or sensory motor or sensory neural pathways that come in through the spinal cord. We've looked at two of those in the previous chapter. One is gracile fasciculus, which is bringing the sensory signals from the lower appendages. The next is cuneate fasciculus, bringing in sensory signals from the upper appendages. So the medulla oblongata is also bringing in the ascending tracts. Two, it contains nuclei. Remember, the nuclei in the brain are specialized regions of gray matter. So there are three nuclei that are present in the medulla oblongata. One is the gustatory nucleus, so sense of taste. These participate in the sense of taste. The cochlear nuclei, cochlea is the organ of hearing, so auditory center is also located here, and the vestibular or the vestibular nucleus. The vestibular organ is also located in our internal ear, and it is the organ, the vestibule of the ear is the organ of equilibrium. So there are these three nuclei that are present in the medulla oblongata of the brain. Quite a few motor functions of the medulla oblongata as well. We already saw how the pyramids contain motor tracts that go down from the cortex of the brain to the spinal cord. Now, the medulla oblongata also contains a cardiovascular center. Now, cardiovascular center is a motor function because it is going to eventually end up to the cardiac muscle. So one, it is going to adjust the rate and the force of how fast or how slow the cardiac muscle contracts. 2, it is regulating the muscles, the smooth muscles of the blood capillaries. So it's not only controlling the heart rate and the force of contractions, but also the blood pressure by dilating and contracting the diameter of the blood vessels. Respiratory center, that is a motor center as well, because it controls the depth of breathing as well as the rate. So how fast we're breathing and how deeply we're breathing. Now the interesting one is the reflex center. We have reflex center in our medulla oblongata as well. So this is what stimulates. The medulla oblongata is stimulating functions like sneezing, cannot control it, gagging, swallowing, coughing, salivation. So these are autonomic responses, if you will. So to recap, the medulla oblongata has sensory functions and motor functions. In the sensory part, it has the 3 nuclei, one for taste, one for hearing, and one for equilibrium. It contains the paths for 2 ascending tracts, the graciofasciculus as well as the cuneate fasciculus. In the motor category, it adjusts the heart rate and the force of contractions. It adjusts the diameter of the blood capillaries, controls rate and depth of breathing, and stimulates involuntary reflexes such as coughing, gagging, sneezing, salivation, that kind of stuff. Now the next part of the brainstem is the pons. So this right here, let me change the color. This right here is the medulla oblongata, and this bump right here is the pons. It's a noticeable bulge in the brainstem, and it contains continuations of those ascending and descending tracts. So this is like a highway. It contains ascending and descending tracts. You have nuclei in this region as well, and they are referred to as pontine nuclei, and they control voluntary motor output. So it's the effector organs in this case are your skeletal muscles. You also have 2 nuclei in this region that coordinate breathing. So the pons is the bulge or the bump in the brainstem. Its major function is continuation of the ascending and descending tracts, and it has nuclei which are commonly referred to as the pontine nuclei. One of those is for control of motor input. The second one is for the coordination of breath. The next part of the brain is the midbrain, and it's right here. It's located right at the floor of the diencephalon. Right in between the midbrain is your cerebral aqueduct. So this connects the diencephalon to the midbrain. So you can see it right here. Now you have 2 regions here. These are called the colliculi, and I'm going to talk about it in just a little bit. So the midbrain is the connection between the diencephalon and the brainstem. You have a lot of specialized structures in the midbrain. So this is the posterior view of the midbrain. It looks like the bottom of a bell pepper if you look at it, this region. And it has those 4 nubs. Together, they are referred to as the tectum or corpora quadrigemina, because there are 4 nubs. The 2 on the superior side are called superior colliculi. The 2 on the inferior side are inferior colliculi. Now the colliculi are centers for reflexes. Now the superior colliculi are responsible for head reflexes to visual stimuli. Now think about this. If you end up catching a movement from your peripheral vision, you tend to turn your head towards the movement. That is being stimulated by the superior colliculus. The inferior colliculus also turns the head, but instead of vision, it turns it to sound. So if you hear a sound, you tend to turn your head towards the sound. So that is stimulated by the inferior colliculi. So superior colliculi and inferior colliculi. Now what you can't see very clearly in this image, you can somewhat see it, are the peduncles, the cerebral peduncles. Now the peduncles are little joints or little junctions that contain descending motor tracts. So they're home to descending tracts. Now when you're looking at the transverse section superior view of the midbrain, you can see the cerebral aqueduct right here in the center. You can see the colliculi right here. And you can see the peduncles as well. So this is a cerebral peduncle, contains a motor tract, and this one contains, not a motor tract, several motor tracts, and this one contains motor tracts as well. Now, in the midbrain, we have a nucleus called the substantia nigra. Now, substantia nigra are these two dark lines right here. And they contain neurons that secrete dopamine. Now, dopamine helps to send inhibitory signals to the motor neurons. Now, degeneration of this leads to Parkinson's disease because there is too much involuntary movement of voluntary muscles going on with Parkinson's. Now the next thing we're going to go over is the reticular formation. So reticular formation is nothing but a loose web, if you will, of gray matter. So it's not a nucleus, so it's not a specialized region. It's a loose web of gray matter that is scattered throughout the brain. And this is, its function is to serve as the reticular activating system. So the reticular formation is part of the reticular activating system, which governs how conscious or how active you are versus how drowsy. So it kind of controls your circadian rhythms. We'll talk about this in just a little bit. So here's a sagittal section, medial view of an actual brain. So you can clearly see the cerebrum. You can see the gyri and the sulci of the cerebrum. So this is a cut right through that longitudinal fissure. So this right here is the medulla oblongata. This is the pons. This right here is the midbrain. So you can see the peduncles right here. This is the superior colliculus. This is the inferior colliculus. The next structure we're going to look at is inferior to the cerebrum and posterior or caudal to the brain stem, and it is this guy right here, the cerebellum. Now the cerebellum sits kind of on top, so if you see the fourth ventricle, it's kind of sitting on top of that fourth ventricle, and it consists also of two hemispheres. So you have a left cerebellar hemisphere and a right cerebellar hemisphere. The cerebellum is very rich in gray matter. So this reddish stuff that you can see, the darkly colored stuff, all of that is the gray matter of the cerebellum. The one in the center, the one that looks like a tree branch, that is the white matter of the cerebellum. Now, the brain, the cerebellum is connected to the brainstem via cerebellar peduncles. You have the cerebral peduncles that connected the brainstem to the diencephalon. You have cerebellar peduncles that connect the cerebellum to the brainstem. Now the functions of the cerebellum are coordination of muscle contractions. So it coordinates, we'll talk more about this when we get to the lab, but it is the coordination center for muscle contractions. So the next section that we're going to go over is the diencephalon. So whatever I'm circling right here is the diencephalon. That is the diencephalon right here. Now this hollow region is the lateral ventricle. The diencephalon has this lighter stained region. It's like a thick sheet. And that is called the corpus callosum. It encloses, the diencephalon encloses the thalamus. The region above the thalamus is referred to as the epithalamus, and the region below the thalamus is called the hypothalamus. hypothalamus. So let's look at the different regions of the diencephalon. The first one is this red bean-shaped structure right here, and that is called the thalamus. It's an oval, very distinct structure, and it's surrounded by the lateral ventricles. The third ventricle separates like the lateral halves. So here, right here is the third ventricle. So it separates the left side of the thalamus from the right side of the thalamus. And the thalamus serves as a relay center. So whatever afferent signals are going to come in through the ascending tracts, they are first going to go to the thalamus. The thalamus is going to decide if it needs to be sent to the cerebellum or the cerebrum or the midbrain. The region below the thalamus, so this brown region below the thalamus, whoops, not there, but below the thalamus is called the hypothalamus. The hypothalamus is a major control center for the autonomic nervous system, and also the endocrine system, because it has the master gland of the body, which is the pituitary gland. So the pituitary gland is housed, or is a part of the hypothalamus. Now the hypothalamus itself has several functions. One, because it houses the pituitary gland, it helps secrete hormones and regulate metabolism, reproduction. So when a woman ovulates, how much testosterone is secreted by a male, all of that is governed by the pituitary gland, which is housed in the hypothalamus. Two, it controls heart rate, things that are part of the autonomic nervous system. So heart rate, breath rate, blood pressure, that kind of stuff. It regulates behavioral patterns. So how angry a person is. If a person is feeling at peace, if a person is feeling aggressive, that is courtesy of the hypothalamus. It regulates hunger and satiety. So when you feel hungry, when you feel full, is controlled by the hypothalamus. Thermal regulation. So it monitors body temperature, and we looked at the thermal regulation homeostasis circuits in our very first chapter. It's a negative feedback loop. So it regulates the body temperature by monitoring the body temperature. And it also controls circadian rhythms. So circadian rhythms are nothing but the internal biological clock. So when it gets dark, you tend to feel sleepy. When it gets bright outside, you tend to feel awake. So that is because of secretion of certain hormones, and the secretion of these hormones is controlled by the hypothalamus. Now the next section is the epithalamus. The epithalamus is posterior, or towards the caudal side of the thalamus, and it is home to a pineal gland. Now pineal gland is a gland that secretes a hormone called melatonin, which is the sleep cycle hormone. It promotes your circadian rhythms and seasonal your seasonal biological clock, puberty as well. So it secretes melatonin. So melatonin is actually a supplement as well. You can find it at a lot of natural food stores. And it's said that if you are traveling across time zones and going to have jet lag, then you should take melatonin at night and try to go to bed, and then try to expose yourself to sunlight in the morning in order to beat that jet lag faster. Now, the epithalamus is also home to a nucleus called the habenular nucleus. Now, the habenular nucleus is interesting because it's not governing. It's not the center for smell. Olfaction refers to the sense of smell. So the habenular nucleus is not a center for the sense of smell, but it links smells with emotion. So this kind of gives you an associated memory that comes with certain smells. So if you went on a first date and if you had roses, every time you smell roses, it could remind you of that first date. So that is smell linked with emotion and memory. The next, or the biggest region of the brain, is the cerebrum. And we've already talked about the longitudinal fissure. We've talked about the frontal lobes, the parietal lobes, and the occipital lobes. We've talked about the central sulcus as well. Now, the whole purpose behind having these gyri and sulci is to increase surface area. Now, over here, what you see in the light pink are the frontal lobes. The blue ones are the parietal lobes. Green is the occipital lobe, and the dark pink is the temporal lobe. Now, enclosed behind or medially to the temporal lobe is another lobe called the insula. So it's kind of shaded right here. So this yellow wing-like structure, that is the insula. What we're going to do now is we're going to look at tracts. You know that tracts are nothing but a bundle of axons that have a similar origin and a similar destination. So we're going to look at tracts that connect different parts of the brain. Now, there are three types of tracts. You can have a tract that connects the left hemisphere to the right hemisphere. So this is the right hemisphere, this is the left hemisphere. So these yellow tracts right here, those are called the commissural tracts. So commissural tracts are tracts that travel through the corpus callosum, and they connect the right hemisphere to the left hemisphere. Now, you can have tracts that stay within the same hemisphere. So they connect different areas within the same hemisphere. The ones up here in the red. So all of these red tracts, those are called association tracts. So association tracts connect one area from the one hemisphere to another area from the same hemisphere. So left to left, right to right. And next are projection tracts. So the tracts here in green are the projection tracts. So they connect the lower cerebral areas to areas within the cerebral cortex. So they project outwards and upwards. So you know that the cerebrum is a large structure. It has gyri, it has sulci, it has those different lobes. So you know the frontal lobes, the parietal lobes, the occipital lobes, temporal lobes, and the insula. You know the longitudinal fissure, and you know the central sulcus. So the three tracts that connect different areas within the cerebrum are one, the commissural tracts. They connect one hemisphere to another hemisphere. So left to right or right to left. Next, you have association tracts. Association tracts go within the same hemisphere. So parts of the left hemisphere are connected to parts of the left hemisphere via association tracts. The third kind of tract is the projection tract. The projection tract connects parts of the lower cerebrum to parts of the cerebral cortex. So from downwards to upwards. So this concludes part one of the brain lecture. In the next one, we will go over the different areas, the association, the motor, and the sensory areas within the cerebrum.